respiration in plants. Respiration is an energy releasing, enzymatically controlled catabolic process which involves a stepwise oxidative breakdown of food substance inside living cells. C6H12O6 plus 6O2 plus gives 6CO2 plus H2O plus energy. 6H2O. Living organisms require energy for all activities like absorption, movement, reproduction, or even breathing. Energy required is obtained from oxidation of food during respiration. Cellular respiration is the mechanism of breaking down of food materials within the cell to release energy for synthesis of ATP. Breaking down of complex molecules takes place to produce energy in cytoplasm and in the mitochondria. Breaking down of C-C bond of complex compounds through oxidation within the cells, leading to release of energy is called respiration. The compounds that get oxidized are called respiratory substrates. Energy released during oxidation is not used directly but utilized in synthesis of ATP, which is broken down when energy is required. Therefore, ATP is called the energy currency of cells. The process of respiration requires oxygen. In plants, oxygen is taken in by stomata, lenticels, and root hairs. Plants get along without respiratory organs because each plant part takes care of its own gas exchange needs. Plants do not present great demands for gas exchange. Distance that gases must diffuse in large plant is not great. During photosynthesis, O2 is released in leaves and diffused to other part of leaves. During process of respiration, oxygen is utilized and carbon dioxide and water is released along with the energy molecules in the form of ATP. Respiratory quotient is the ratio of volume of carbon dioxide produced to the volume of oxygen consumed in respiration over a period of time. RQ is equal to 1 for carbohydrate and less than for protein and peptones. Respiration, aerobic and anaerobic respiration. Aerobic respiration, glycolysis, Krebs cycle, terminal oxidation. Aerobic respiration is an enzymatically controlled release of energy in a stepwise catabolic process of complete oxidation of organic food into carbon dioxide and water with oxygen acting as terminal oxidant. Glycolysis. The scheme of glycolysis is given by Gustav Amden, Otto Meyerhoff, and J. Parnas. It is also called as EMP pathway. Glycolysis is the partial oxidation of glucose or similar hexose sugar into two molecules of pyruvic acid through a series of enzyme-mediated reaction releasing some ATP and NADH2. It occurs in cytoplasm. In plants, glucose is derived from sucrose or from storage carbohydrates. Sucrose is converted into glucose and fructose by enzyme invertase. Glycolysis starts with phosphorylation of glucose in presence of enzyme hexokinase to form glucose 6-phosphate. One molecule of ATP is used in this process. In next steps, glucose 6-phosphate is converted into fructose 6-phosphate catalyzed by enzyme phosphohexose isomerase. Fructose 6-phosphate uses another molecule of ATP to form fructose 1,6-biphosphate in presence of enzyme phosphofructokinase. Pyruvic acid, lactic acid fermentation, alcoholic fermentation, aerobic respiration. In glycolysis, two molecules of ATP are consumed during double phosphorylation of glucose to fructose 1,6-biphosphate. Two molecules of NADPH2 are formed at the time of oxidation of glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. Each NADH is equivalent to 3 ATP, so that net gain in glycolysis is 8 ATP. Pyruvic acid is the key product of glycolysis. Further breakdown of pyruvic acid depends upon the need of the cell. In animal cells, like muscles during exercise, when oxygen is insufficient for aerobic respiration, pyruvic acid is reduced to lactic acid by enzyme lactate dehydrogenase due to reduction of NADH2. In fermentation by yeast, pyruvic acid is converted to ethanol and CO2. The enzyme involved is pyruvic acid decarboxylase, and alcohol dehydrogenase catalyze this reaction. In both lactic acid fermentation and alcohol fermentation, very less amount of energy is released. Yeast poison themselves to death if concentration of alcohol reaches above 13%. Final product of glycolysis, pyruvate, is transported from cytoplasm into mitochondria for further breakdown. Oxidation of pyruvate to acetyl CoA is done to produce CO2 and NADH. The reaction catalyzed by pyruvic dehydrogenase requires the participation of several coenzymes including NAD+. Pyruvic acid plus CoA plus NAD plus in the presence of Mg2 plus pyruvate dehydrogenase gives acetyl CoA plus CO2 plus NADH plus H plus. The acetyl CoA enters a cyclic pathway called TSA cycle or Krebs cycle. Tricarboxylic acid cycle, Krebs cycle. TSA cycle was discovered by Hans Krebs in 1940. This cycle is called TSA cycle because initial product is citric acid. Acetyl CoA combined with OAA, oxaloacetic acid, and water to yield citric acid in presence of enzyme citrate synthase to release CoA. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation, leading to the formation of alpha ketoglutarate acid and then succinyl CoA. In the remaining steps, succinyl CoA is oxidized to OAA, allowing the cycle to continue. There are 3 points in the cycle where NAD plus is reduced to NADH2 and 1 point where FAD is reduced to FADH2. A molecule of glucose produces 2 molecules of NADH2, 2 ATP and 2 pyruvate while undergoing glycolysis. The 2 molecules of Chapter 14. A molecule of glucose produces 2 molecules of NADH2, 2 ATP and 2 pyruvate while undergoing glycolysis. The 2 molecules of pyruvate are completely degraded in Krebs cycle to form 2 molecules of ATP, 8 NADH2 and 2 FADH2. Pyruvic plus 4 NAD plus FAD plus 2H2O plus ADP plus PI. In presence of mitochondrial matrix, 3CO gives 3CO2 plus 4 NADH FADH2 plus 4H plus ATP. Terminal oxidation is the name of oxidation found in aerobic respiration that occurs towards end of catabolic process and involves the passage of both electrons and protons of reduced coenzyme to oxygen to produce water. Electron transport chain. The metabolic pathway through which the electron passes from one carrier to another inside the inner mitochondrial membrane is called ETC or mitochondrial respiratory chain. Electrons from NADH produced during citric acid cycle are oxidized by NADH dehydrogenase and electrons are transferred to ubiquinone located within the inner membrane. Ubiquinone also receives electrons from FADH2, which is transferred to cytochrome C via cytochrome BC1 complex. When the electrons pass from one carrier to another via electron transport chain, they produce ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends upon electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while oxidation of one molecule of FAD2 produces 2 molecules of ATP. Oxidative phosphorylation, it occurs in respiration process. Photophosphorylation, it occurs in photosynthesis. Energy of oxidation reduction is used for production of proton gradient required for photophosphorylation. Light energy is utilized for production of proton gradient for phosphorylation. The energy released during ETC is used to make ATP with the help of ATP synthase, which consists of two major parts, F1 and F0. F1 is a peripheral membrane protein complex having site for synthesis of ATP from ADP and inorganic phosphate. FO is integral membrane protein that form channel for proton. For each ATP produced, 2H plus passes through F0 from the intermembranous space to the matrix down the electrochemical proton gradient. Fermentation. It accounts for incomplete oxidation of glucose. Aerobic respiration. It accounts for complete oxidation of glucose. In fermentation, there is net gain of only 2 molecules of ATP. In aerobic respiration, there is more net gain of ATP. NADH is oxidized to NADS plus very slowly. NADH is oxidized to NAD plus very fast. Amphibolic pathway. Glucose is the favored substrate for respiration. All carbohydrates are usually converted into glucose before used for respiration. Fats needs to be broken down into glycerol and fatty acid, which is further converted into acetyl CoA and enter the respiratory pathway. Proteins are broken into amino acids and further enter into Krebs cycle. Breaking down process within living organism is called catabolism, and synthesis process is called anabolism process. So respiration is an amphibolic pathway. Done.